The American Society for Microbiology has a special offer for listeners of ASM podcasts. Get 20% off one of our favorite books, The Invisible ABCs, using promo code ABC20. That's ABC20, one word. The Invisible ABCs, Exploring the World of Microbes, will delight readers of all ages with its colorful presentation and spectacular selection of illustrations. Intended for school-age children and younger, this unique book will stimulate parents, teachers, librarians, and even older students to discover the fascinating world of microorganisms. And if you're a current ASM member, you receive this discount for all titles in our catalog. To check out The Invisible ABCs and take advantage of this special before it's gone, visit asm.org/books. That's asm.org/BOOKS. Or you can simply click the link in the description for this episode in your podcast app and use promo code ABC20 at checkout to get 20% off this collection of over 250 superb illustrations, including algae, mold, bacteria, protozoa, and viruses that present scientifically accurate information in an age-appropriate format that is both colorful and captivating. Give the gift of microbiology to a new generation of microbial explorers at asm.org/books.
This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 216, recorded on April 23rd, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hello, everybody.
Sound great.
Good.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, everyone.
And from Charleston, South Carolina, Michael Schmidt.
How's everyone today?
And we can all wish Elio a happy birthday, right?
Yeah.
Prematurely, but thank you.
It'll be Sunday.
It'll be Sunday. That's
and when this episode is released, it'll have been passed, and we will have wished you so many, many more.
Thank you so much. I appreciate it.
Are you staying well during the pandemic?
I'm doing very well. Yeah, I'm totally okay. A little bit
All
bored, but other than that, I'm totally okay.
Oh, if you want to make more podcasts, we can do that, and then so you won't be bored.
Okay.
Or you can just listen to them.
That's true.
I wanted to give everyone a link to a lovely New York Times Magazine article, which, of course, will be out this Sunday, but it's already online. It is by Jennifer Kahn. It's called How Scientists Could Stop the Next Pandemic Before It Starts.
Oh,
and
wow.
It features me and a couple of other people.
Hey.
So I'm being self-serving here. I'm sorry, but she did a great job of
self-serve away.
So that's very cool.
And is it focused on interventions, non-therapeutic interventions, all of the above?
Well, it's a focus on what could we do to prevent this next time. And one is to have therapeutics ready, which we could have had for this, but there were, monetary considerations as we discussed. And then to do more wildlife sampling, to know what viruses are out there and which are pandemic threats, which is very hard to get funded. And so she also has Peter Daszak, who's the head of Eco Health Alliance, which funds a lot of these expeditions to do field sampling of bats and various other critters around the world to see what viruses they have in them. But anyway, she really understood it. And I worked with a fact checker for the Times, and she said she agreed that she did a great job in writing the science, which is not always easy for a writer to do.
Well, that's great. I look forward to it.
I'll put a link in the show notes. If you get the magazine, it'll be glossy.
All right.
With pictures?
Well, not of me, no, but
not
there's
the
just
famous
one
polio wall?
No pictures, but they have a graphic, kind of a cool graphic in the beginning. Anyway, I just wanted to point that out.
Well, we're lucky to have you getting the word out, Vincent. Thank you.
Well, I've been responding to all calls, frankly, and I don't say no. Yesterday morning at 7:30 here Eastern, I was on Beijing television.
Wow.
Which was really cool because it was a regular TV studio and they piped my Skype video into it. Incredibly professional people, all fluent, perfect English, and really nice. And of course, they wanted to dispel this idea that China made this virus, which, won't die.
It's awful.
And I was very, very happy to say, no, it was not made by any lab anywhere. It came from nature, and please, let's move on. I feel badly because the anchor said to me he has friends in the U.S. and they fear for their life because they're afraid that being
God.
Chinese now they're going to be attacked. Isn't that horrible?
That is.
There's so many examples throughout human history of pathogens jumping from animals to people.
Like you said last time on TWiM, right?
Yeah.
Then salmonella, neolithicization.
Right.
Yes. And so what it is that people don't really know much about history and they ignore it and make up their own ideas. But I think, as Alan Dove said on a TWiV, it's mostly political. They just want to put the blame on another country
even
for
the
something
flu
they didn't do.
virus, every year it emerges due to mixing, right?
Well, after all, that's how Spain got blamed. They were the only ones that had a free press at the time. The rest of the globe was engaged in a world war, and Spain was the only country not. And
that's true. That's absolutely true.
It was Spanish flu, and it had nothing to do with Spain. It probably came out of Kansas, as we now know, the great flyway of the Midwest. And what we have is crazy.
Now it's called Spanish flu, and it's not going to go away, right? The name will stick forever.
The name will stick.
All right, so I wanted to do two quick snippets, all dealing with SARS-CoV-2, but quite interesting. And the first, very brief, is an announcement from the NIH yesterday. It's called Treatment Guidelines, COVID-19 Treatment Guidelines. And this is a treatment guidelines to inform clinicians how to take care of patients. And the idea here is that the management is evolving because the clinicians are learning new things. So they're going to update these, and they're based on evidence, scientific evidence and expert opinion. And they have two ratings, a letter that indicates the strength of the recommendation, A, B, or C. And you can imagine what A means, right? And what C means. Mm. And a Roman numeral 1, 2, or 3 that indicates the quality of the evidence that supports the recommendation. And this is a very august panel of mainly clinicians from many different places, from various medical centers, NIH, and medical centers throughout the U.S., and all with, there's some PhDs there as well for good reason. So this is a website which we'll put in the show note, and they have a very nice explanation. And so here at the moment, here are the summary recommendations. Very interesting. I'm just going to go through a couple of them. The treatment guidelines does not recommend the use of any agents for pre-exposure prophylaxis against SARS-CoV-2 outside of the setting of a clinical trial. And
that may change, right?
It may change, absolutely. But I think this is trying to, absolutely hope. This is trying to ward off, someone saying, hey, everybody take this drug. It's going to save the world, right?
Right.
Would anyone do that?
Yes. People are desperate.
The panel does not recommend the use of any agents for post-exposure prophylaxis outside the setting of a clinical trial. That gets an A3. The other one got an A3. Third, the panel recommends no additional laboratory testing and no specific treatment for people with suspected or confirmed asymptomatic or pre-symptomatic SARS-CoV-2 infection. Okay, so if you have a test and it's positive and you don't have any symptoms, then that's it. Nothing else to do. And finally, no drug has proven to be safe and effective for treating COVID-19. There are insufficient data to recommend either for or against the use of any antiviral or immunomodulatory therapy in patients with COVID-19 who have mild, moderate, severe, or critical illness. So that's a summary of their main points. And
that's sobering, isn't it?
Yes. And
so all the excitement about the IL-6 inhibitors have not yet panned out.
there's anecdotal evidence. Like Daniel Griffin says, I gave this patient a second dose, and the next day we could reduce the oxygen to 70%. I don't know what that means, right?
No one does. And that's why they're doing evidence-based practice. They are effectively citing the grade of the evidence, the number of patients in it, whether it was a randomized controlled trial, all of the things we teach our students about evidence-based support before you make a clinical decision have to be followed if we're going to come up with the secret sauce.
But at the same time, we absolutely need our physicians to be, observing and sharing what they see, because that's where we get ideas, right? It just then needs to be done in a systematic, controlled fashion, collect that information.
Totally. And that's one of the shameless plugs I'll give for TWiM. I mean, I look forward to Sunday mornings when I get to hear Dr. Dan Griffith share his observations from his week's experiences in Colombia. I mean, they have been really profoundly informative, and really, I've heard from a number of clinicians that I've turned on to the TWiM podcast. They listen religiously to what Dan has been seeing in Colombia, because, New York City is ground zero for the bulk of the disease that we're seeing in the United States.
We're very fortunate to have him every Friday. He goes out to his car and he gets on his phone for half an hour and he tells us what's going on clinically.
Yeah.
And so, go over to TWiM and listen to that. Even if just for that first half hour, it's very useful to hear how things are evolving from week to week. It's very interesting. Things change. And he says it. He says, well, we've got something new this week. We had to put some patients on kidney dialysis.
Yeah.
Like the virus might be infecting kidney. So he said, we have so many patients now. That's the thing. You're going to see all kinds of minor happenings that you wouldn't normally see when you have fewer patients.
Right.
And, he's effectively 1 week ahead of the national news. Many of us heard last evening that kidney dialysis was a reality for many COVID patients, and he informed us of this last week. So you can hear it there first before you have to wait for the evening news.
You bet.
Yeah,
I think
he makes it very accessible to a broad audience. I'm with you, Michael. I really look forward every Sunday to hear what the latest is from the clinic. And it also reminds me just how hard our clinical colleagues are working, and that we all need to do our part by practicing social distancing and other measures to keep the numbers of patients low.
Yeah, it's some states are talking about opening up already, and Daniel says, don't do that. No. We're just starting to get our necks above water.
That would be my neighbors to the east or the west. I have to remember where Georgia is.
There's nothing to the east of you, Michael. Right?
No, water. We have lots of water. We have lots of water. Okay.
The other, the real snippet I want to do is a paper Michael sent me out of Yale, and it's a medRxiv preprint. But to me, it looks pretty good. It's called Saliva is More Sensitive for SARS-CoV-2 Detection in COVID-19 Patients Than Nasopharyngeal Swabs. So this is for...
That's a big deal, isn't it?
Oh, it is a big deal. I mean, think about having a swab that requires finesse to be placed almost to your brainstem, and then you cough and sneeze on the person collecting the specimen, versus you turning your head and just expectorating some saliva into a cup.
You don't have to expectorate, you just spit.
Yeah, well, we can say spit, I suppose.
Sure. Yeah, and plus, it gets around swab shortages, right?
Oh,
for other countries, and so forth. Saliva is very, very interesting. So they say saliva has a lot of advantages, but it hasn't been rigorously evaluated. And so that's what they did in this study. And the bottom line is saliva is more sensitive and consistent than using nasopharyngeal swabs. And this makes sense.
They're very busy at Yale, aren't they? A lot of stuff is coming out of Yale.
Yeah, many people are working at many places. It's amazing. Sure. It's just great.
But I agree, Vincent. It makes sense because we're seeing transmission, pretty easy transmission, and you'd think that it would be coming out of our mouth, not just our nose, huh?
Sure. And then, saliva and, your nasopharyngeal cavity, oropharyngeal cavity, it's all continuous. So
right.
If you have virus in the epithelium, it's going to be in saliva. It's not surprising. Yep. But they needed to do this formal study. They have 44 patients, COVID-19 patients, with a range of severities. And they have 121 self-collected saliva or healthcare worker administered nasopharyngeal swabs, right? So you can take your own saliva sample. You don't have to have someone put a swab in your nose. And as Michael said, it's very uncomfortable if it's done right. And
so then we really could ramp up to broad testing.
Oh yeah,
wide testing, which
you don't have a sampling issue where you have to go somewhere. You could just send in saliva, but
have it show up in our mailbox and then we ship it back.
Oh, wouldn't that be brilliant?
Yep.
Oh yeah,
I have this box here that I got 3 years ago from NYU study. They were doing a respiratory virus scan. I enrolled, but I never did it. It came with a nasopharyngeal swab. I'm supposed to do it myself.
Oh God!
and a little tube of medium. I think it's very hard to do because you're going to start gagging and pull it out, right? Anyway, I still have it. I should just keep it as a souvenir for the future. So then they took these swabs and they compared them. So they have the saliva and nasopharyngeal. They do PCR, and they find the RNA levels, which is what you're measuring here. So they call it virus titers, but that's not correct.
Yes.
My colleagues from Yale, it is RNA level, because it's not even a genome, because they could be broken pieces of RNA. 5 times higher in the saliva than in the nasopharyngeal swab. So higher.
So this is why we're doing this paper on TWiM. They won't get yelled at by all the virologists on TWiM.
Yeah, maybe they're not listening anyway, right? And then they tested longitudinal samples to see which are more consistent. And they have 22 people here. And the RNA titers generally go down in both samples. But in 5 cases where a nasopharyngeal swab was negative, the saliva was positive. And there was no instance where a sample was negative and then positive with saliva, whereas that happens all the time with PCR. So not only more sensitive, but more consistent than the nasopharyngeal swab. And then the last thing they do is look at subclinical infections. And so far they found they have found RNA in saliva from 2 healthcare workers who were negative by PCR by nasopharyngeal swabs. All right, so this is much more sensitive. So that's it basically. And they say properly at the end, we need to do a larger sample. This is only 122 patients, but I think the data look good, and I think that we should start doing this.
So it sounds like they haven't done plaque assays yet to know if there are live virions in the saliva.
No, they haven't. Yeah. They haven't. No.
There's an early paper out now, albeit it's based on the first iteration of the coronavirus SARS, and it shows that SARS virus does indeed replicate in the epithelial cells that line our salivary ducts. And so it's probably infecting that same tissue, and their virus will probably be recoverable. But again, that requires you to have a cell culture set up, a biosafety level 3 apparatus, whereas PCR, of course, you can get away with after you inactivate the virus through extraction of the RNA. You can get away with it in far more laboratories than a viral titer assay, which will make infectious virus.
So that was an older study done with SARS-1, or is it a recent study?
Hold on a second. I'll dig out the paper here.
You actually have a printed paper, Michael?
I print papers because it's faster than my computer. So it's from the Journal of Virology, April 2011. Epithelial cells lining salivary gland ducts are early target cells of severe acute respiratory syndrome coronavirus infection in the upper respiratory tract of rhesus macaques. And it was a group out of the University of Hong Kong, and it was published in the journal Virology in April 2011, 9 years ago. Volume 85, issue 8.
It's a different virus.
Different virus, yeah.
Well, but the target is, epithelial cells and...
Yeah, but you still need to repeat it.
Sure. No arguments.
Anyway, that's saliva for you. So, Michele, what do you have for us today?
Yeah, I have a paper that is, I think, relevant to today's epidemic of coronavirus. It is entitled Mycobacterium tuberculosis sulfolipid 1 activates nociceptive neurons and induces cough. Was published in Cell this month by a group from University of Texas, Southwestern Medical Center, and also University of Texas at Dallas. The authors are Cody Rule, Brianna Pascoe, Harris Khan, Lexi
You're going to mention more? My God, we're going to be here a while.
I'll go fast. Chelsea Stem, Luis Franco, Connie Shea, Min Zhao, Colton Davis, Tien Chen, Laurent Graton, Michael Burton, Kalo Ohia, what you said, and Don and Jay Niek, also Gregory Dresser, Theodore Price, and the senior author, Michael Shiloh. So the summary here is that Mycobacterium tuberculosis is an ancient and highly successful pathogen, but it is utterly dependent on humans for its existence, to replicate in our lungs and then spread to the next host by coughing, and then the next person inhales those contaminated respiratory droplets. Does this sound familiar?
It sounds exactly what we were just talking about.
Yeah. So here, Cody Rule, Michael Shiloh, and their collaborators discovered that this bacterial pathogen releases a factor that makes its host cough, thereby promoting spread of the bacteria to the next innocent bystander. So I thought this was amazing. Now, for the history of Mycobacterium tuberculosis, this disease was described by ancient Greeks and Romans, and it's now been immortalized in our more current films and operas. But it actually dates back 2,000 to 3,000 years BC, where mummy spines have evidence of disease caused by tuberculosis. So it's now estimated that roughly a third of the world's population actually carries TB in our lungs, where it can persist within these walled-off structures called tubercles. And then if your immunity weakens as you age, or you're on an immunosuppressive therapy, or you have uncontrolled HIV infection, these zombie bacteria can awaken, replicate, and then cause serious infectious pneumonia. So the authors were motivated to do this study because they thought, this ancient, highly successful pathogen, it's completely dependent on us to replicate and spread. Would they leave that to chance? Instead, they bet that no, it's more likely that over the eons of coevolution with humans and MTB, that the bacterium acquired tools that intentionally tickle the human cough reflex. So that's what motivated this amazing study. So cough, we think of it as, we probably don't even think of it at all. We take it for granted. But I want to quote a review from Stuart Brooks in 2011, who writes, "The cough reflex is a multifaceted, precisely timed neuromuscular phenomenon characterized by the precise, concurrent, and sequential coordination of the activation patterns of the diaphragm, various muscle groups of the chest wall, cervical muscles, abdominal muscles, laryngeal, abductor and adductor muscles, medullary and higher cortical regions of the brain, et cetera, et cetera." So it's quite
I thought I was just coughing.
Yeah. So if they want to study, does Mycobacterium intentionally trigger cough? Of course, they need an assay. They need a cough counter, and they need an animal model. So the figure 1A shows this really elegant small chamber where they can put a guinea pig and it can move about freely. And then they can introduce a nebulized solution, so they can create mists that are contaminated with their agent of choice. And then the instrument also has a pressure sensor. So you can imagine as a guinea pig or a person is coughing, they're changing the pressure in the chamber. And this device can sense that and plot it on a graph. So their first challenge was to get this assay up and running. So they had a positive control, a molecule called capsaicin that is known to trigger this response. And of course, a negative control is easy. But then isn't
that the stuff that's in hot peppers?
Yeah. I think it is.
Yeah, and we've all experienced that going to Stan Malloy's favorite Mexican restaurant, where he gives us the very hot peppers, and then we all cough for the next 20 minutes.
So then they added to this nebulizer lipid extracts from Mycobacterium tuberculosis. So why lipid extracts? It's well known that MTB has a very greasy outer membrane, lots of complex lipids, and some of them have been studied and known to have biological activity. So they thought this is a great place to start. So they prepared extracts from MTB, put them into their nebulizer, and eureka, the guinea pigs coughed. So, Michael, I'm making this sound all very easy and elegant, but would you take us behind the figure and tell us about what Cody's experience was getting this assay up and running?
Oh, well, this was one of his biggest moments, just getting the cough experiments to work. Now, you have to appreciate them working with Mycobacterium tuberculosis. You're already in a biosafety level 3 containment lab. And so he had uninfected animals, he had wild type animals, or he had uninfected, wild type knockouts and complement strains of the various forms of Mycobacterium tuberculosis. And then he infected them into these guinea pigs. So just imagine one guinea pig in what effectively looks like a bell jar that has a pressure transducer at the top, and you have to make up the solution, and then you have to have a nebulizer, which effectively vaporizes the liquid that you have prepared. It was just crazy. So Cody, just the actual setup and troubleshooting of all the 24-hour cough experiments took us over a year. And that was just for showing that the guinea pigs infected with the wild type extract of Mycobacteria could cough, and expanding to do the mixing and matching experiments that Michele is going to take us through. You can well imagine that took a long time to get set up. But then in order to do the viable counts, tuberculosis grows very slowly, and that takes another 2 to 3 months in order to get the data. So by the time they got up to, there are 5 figures in this paper, by the time they got us to figure 5, both Cody and the senior author were extremely happy and relieved.
Yeah, it took a lot of fortitude. And I'll just add, this is why we call it research and not search. It takes painstaking work to develop these assays. So having gotten this clue that, in fact, the greasy lipids that are shed by virulent TB does induce a cough, of course, they then wanted to get at the mechanism. And to do that, they wisely switched to an in vitro model looking at neurons that are known to transmit this signal for coughing.
By the way, I looked up what nociceptive means. Noci means pain, that the neurons that deal with pain reception.
Great.
And so Cody had experience in neurobiology, and he came, if you will, pre-selected to work in Dr. Shiloh's laboratory. And so he was already primed to do this assay that Michele is now going to describe.
Wow, a great combination. So they started with a cell line from mice of dorsal root ganglion cells that are known to transmit this signal. And they knew from the literature that when these cells are stimulated, they take in extracellular calcium. So the intracellular calcium rises rapidly. And fortunately, we have fluorescent dyes that bind calcium, and we have imaging methods, microscopes, and the accompanying video cameras and software where we can get traces of the calcium fluxing in. And that is a readout then of the activation of these nerve cells. So they set that system up and then treated the nerve cells either with capsaicin, which is known to trigger the response, and they got that positive control to work, or they treated it with vehicle alone and got no signal, or they added the MTB extract to these neuronal cells. And again, Eureka. Figure 2 shows really beautiful quantitative data over time that really convincingly shows that something in those extracts from the greasy outer membrane of MTB, in fact, was inducing these neuronal cells. They then verified that it wasn't unique to just this one mouse cell line. They also used a number of other different type of mouse neuronal cells, and they made fresh human dorsal root ganglion nerve cell cultures and found that the same response occurred there. So now they're excited and they're motivated to try to understand what is the secret sauce in all these greasy lipids that they've extracted from MTB. So their first step in that was to take advantage of a known collection of mycobacterial species, some that cause human disease and some that don't, and ask whether they too would trigger this calcium influx or trigger these nociceptive receptor on neuronal cells. So that data is shown in Table 1, I believe, possibly Table 2. And what they were incredibly fortunate to find is that they had a pair of MTB strains, one a virulent one called the Erdmann strain, and one an attenuated or less virulent strain. It's called H37RA, which is known to not grow as well in animal models or in white blood cells. They also knew it didn't have key two-component regulator system, FOP system, that is required to produce one of the major greasy cell lipid wall lipids called SL1. So they, when they used that pair of wild type and mutant Mycobacterium extracts, they found, lo and behold, that the extract from the mutant, in fact, did not trigger the neuronal response. So this was a great lead then, because this lipid that's missing in their mutant, SL1, is very appealing for a number of reasons. First, it's a lipid that was first described way back in 1959 by a physician and TB expert, Gardner Middlebrook, who people may have heard of through Middlebrook medium, which is still used to culture TB. But it's a very abundant sulfated glycolipid. It makes up, yes. Yeah. It makes up 1% of the dry weight of Mycobacterium, so there's a lot of it. It's not made by the environmental mycobacterial species that don't cause pulmonary disease. So they were motivated to obtain purified SL1 lipid, and sure enough, they found that it was sufficient to trigger this calcium efflux in the nerve cells. And that's shown in Figure 3A. Just 33 nanomolar of the lipid induced the half maximal response. So what is this SL1 lipid? The structure is shown in Figure 3B. At its core, it's 2 linked sugars, and they're modified by a sulfate group and then 4 extended fatty acyl chains. Now, they learned that the response was specific to this SL1 because they tested other lipids that they purified from MTB, and they did not trigger the calcium influx of the cultured neuronal cells. They did see one interesting exception, Mycobacterium avium, which does cause respiratory disease in people, especially immunocompromised people, and it also produces complex glycolipids, but not SL1. So it's possible that it has evolved a similar pathway, but by independent mechanism. But we'll get back to that later. So they wanted to understand then what part of the molecule of this SL1 lipid is required to trigger the cough. And here, for these structure function studies, they were able to go to the literature, and there was really a large body of work understood about the biosynthetic pathway for this complex lipid. And these were led by Joseph Mougeot, who's now a professor at University of Washington. But he did this work when he was a graduate student at UC Berkeley with Carolyn Bertozzi, who's just a really talented chemist who's focused on glycolipids and glycosylated species on the surface of cells. So by going to the literature, they were able to identify a whole series of mutants that were blocked at each progressive step of the biosynthetic pathway. And they extracted lipids from each of these mutants and then put them to their test using their neuronal cells and looking for the calcium firing using fluorescence microscopy. And what they found was that the smallest unit that was sufficient to trigger a response, this neuronal response, was the trehalose disaccharide modified with a sulfate group. So it didn't have as much activity as the full lipid, but still it was active. And likewise, when they obtained or chemically synthesized just that precursor molecule, they found it too had activity. They also verified that this lipid was sufficient to stimulate both human and, as I said, the mouse, other types of mouse cells. So they're pretty sure that they've got now a good handle on what the active component is in their extracts, at least in tissue culture neuronal cells. Now they want to really push this and go back to their guinea pig and ask, is this lipid fragment that can trigger calcium influx in their cultured neurons able to actually trigger a cough, this complex behavior in an animal? So again, they went back to this chamber where they put live guinea pigs and created mists that contained either extracts from wild-type Mycobacterium, and yes, the guinea pigs coughed, or they had them inhale lipids from each of the biosynthetic mutants, and they did not, or they prepared the complemented mutant, and it did trigger coughing. So this is the in particular mutant they used was in the very first step of the biosynthetic pathway in the enzyme that adds that sulfated group onto the disaccharide. So this is really exciting, but they pushed it even one step further. So the icing on the cake, they decided to actually, rather than just have the guinea pigs inhale these extracts, instead they infected the guinea pigs with either the virulent strain of MTB or one of the mutants. And then 6 weeks after infection, they quantified how many colony-forming units of either wild-type or mutant bacteria were in the lung. They also did pathology and looked at inflammation as well and found that, again, those were similar whether or not the Mycobacterium made this lipid SL1. However, the only animals that coughed were the guinea pigs that were infected with MTB that had decorated their surface with the SL1 lipid. And that's shown in figure 5.
And they did that before killing the animals to look at the pathology.
Right. That's right. They chose their time points wisely and their dosages wisely.
And the other thing that they didn't talk about, but is germane to our discussions today, is that by not coughing, the infectivity of the mutant strain will likely go down, or its R0 value will be reduced. So it's unlikely that the guinea pig would be able to transmit the mutant Mycobacterium to another guinea pig. So the cough reflex is actually very important for the further pathogenesis of the microbe MTB.
Well, for the transmission, for it to be maintained in the population.
Yeah.
Yep. What's cool is that it's not affecting the pathogenesis in this particular, infected individual. But as you point out, Michael, they're most likely have identified how this pathogen is transmitted so successfully to other people. So this also solved a puzzle that's been long-standing in the TB field. As I mentioned, this lipid SL1 is very abundant. It's known to be a component of virulent strains for more than 60 years. Yet, people have made mutants of TB that lack this lipid. They're still virulent in individual animals, and they actually do better than wild type if you put them in cultured human macrophages. So it had been some question about, why was this retained? Why was it so abundant? Why is it present in the pathogenic strains? And yet we can't, as experimentalists, put a handle on it. And that's because so much of pathogenesis is really focused on growth, infection and growth in one individual. We don't have as good assays for studying transmission, especially as Michael was saying, for MTB, it's highly infectious. To work with it in the laboratory and in animal models, you need a special facility, BSL-3 facilities, to keep yourself safe. So the authors, of course, are interested in doing those experiments, but that will be a big investment of time.
And for TB, the infectious dose is only 10 microbes. So if you cough out more than 10 microbes, and remember, TB can persist on surfaces because of its waxy coat. It's resistant to desiccation. I mean, you can now begin to see why planet Earth has a third of its population infected by this very clever pathogen.
And it can also, because of its waxy coat, stay suspended in tiny droplets, aerosol droplets in air that can stay suspended longer and travel further. So if you walk into a room that somebody had coughed in and left, you could be at risk of infection. So because humans are the only known reservoir of MTB, and we know that it's spread by droplets, this really gives us a new way to think about how we can protect people from this disease. And wouldn't it be great if, rather than having to use social isolation or N95 masks, if future studies could identify the particular receptors and pathways that trigger us to cough, and then develop therapeutics that can suppress the cough response in somebody who's ill, so that we can thereby protect their innocent bystanders that they live with or interact with at the grocery store. So I think this discovery by Cody Rule, Michael Shiloff, and their many talented interdisciplinary teammates is going to really stimulate a broader field of mechanisms of transmission by respiratory pathogens.
And that's one of the notes that Cody says, note for his junior colleagues. On the research side of things, stick with it. Most experiments don't yield the data you're expecting. The important thing is to learn the right questions to ask, as well as learning as many techniques as possible and how to interpret the data correctly from those techniques. Once you're confident in these techniques and knowing which to do and when, you'll be more prepared during graduate school. And he really credits his undergraduate experience in the second Eddy lab at Texas A&M as really priming him to enter the laboratory at UC Southwestern in Michael Shiloh's lab. I mean, that really prepared him for doing such outstanding work here.
Let me get backtrack a little bit. Nobody says that the coronavirus has the same clever skill, does it? We don't know that coronavirus induces cough.
Sure. No, I think I was looking just now for viruses in general, whether we know what causes the cough reflex. And in most cases, it's the inflammatory response of the host, that irritates the respiratory epithelium, and that causes the cough. And so I was wondering here, though, Michele, it looks like it's entirely SL1, right?
Yes.
No contribution for inflammatory response. Is that correct?
It's certainly, we don't know what dosage would be required, and it may be more complex, but certainly their data convincingly show that it is necessary and sufficient to trigger the cough in their guinea pig model.
So the mutant that doesn't make SL1, they don't cough at all, right?
That's H37RA. No, it's very few.
Right. Although, we have to appreciate that every assay has got a limit of sensitivity. So they're not detecting the cough reflex.
So it's very different. Of course, here the bacteria is full of stuff, right? All kinds of molecules. And one of them does this. Whereas viruses, they have to get into cells and make things.
Right.
And so you would have to postulate that something made in an infected cell is coming out, which is not unreasonable, but no one has ever really, maybe this will stimulate people to look at that. It's is this the first time people have shown a bacterial component can induce cough?
It's the first that I am aware of, and it's the first that the authors are aware of also. Yeah, now there is precedent for bacterial products triggering neuronal signaling. I think pain receptors. We talked about a paper, I believe, on Staph aureus here on TWiM.
Indulge me for a moment on looking at things the other way around. On the one hand, this paper says bacteria, TB, teach people how to cough, and thereby they spread to other hosts. The opposite tends to be true too. I once wrote a little piece about this. When people are infected with a common flu virus, what do? Do they become sociable? Not at all. They want to go to bed. They don't want to talk to anybody. What kind of mechanism is that? It's the opposite mechanism. It's the avoidance of spread. So we have, on the bacteria learn how to spread, people learn how to resist that.
That's right. And that's a cytokine response, right? I believe one of the IL something or other triggers us to just feel achy and want to lay down, so we're less apt to spread it to our species.
So what we consider to be a painful thing, namely feeling bad and feeling miserable like we do with the cold, is actually a protective mechanism.
For the species. Yeah.
For the species.
Cool.
So biology is wonderful, isn't it?
It's an interesting way of looking at it. You know, the way I look at it, at least for viruses, is that if they make you too sick, then you're not likely to transmit as well, right?
That's right.
Especially if you die very quickly, like Ebola virus patients.
Right.
But that's why SARS-CoV-2 is amazing, because 80% of infections are mild, right? And people are just walking around spreading that virus. And SARS-1, over half of the infections were serious enough to require hospitalization, and that's why that one was easier to contain. So there is something there for sure.
Yeah, I think a principle that we learn in infectious disease is that as a pathogen co-evolves with its host, it actually dampens its immune response. So the common cold virus is wildly more successful than, say, MERS or SARS, because people are up walking around and they spread it.
Yeah, I mean, I looked up some papers on rhinoviruses, right? And I found one here from a few years ago. How does rhinovirus cause the common cold cough, which they argue is needed for transmission? I think it's part of transmission. So for viruses, it's not just coughing, it's just speaking. And I don't know, what about tuberculosis? Can you spread it by just talking, or does it require a cough?
The infectious dose is so low, and
speaking should do it.
And speaking will do it.
Yeah. You can gram-stain sputum samples, and there are bugs there, right? So
so speaking should be enough. Yeah. So I'm not sure that would be an interesting experiment to see if you don't cough, do you still transmit?
Right.
And I'm sure you would.
So one thing that I was so impressed by with this paper is that they use bacterial genetics, some heavy-duty chemistry, and some use of biochemical extraction of lipids from the cells, and then also the fluorescence microscopy, the animal models. Who was this team, Michael, that put all this together?
It was an integrated group of folks. First and foremost, it's Lexi Kent. She started this project before Cody arrived in the lab, and she helped him transition into working on the project, and she set up the initial calcium imaging and cough experiments. And then doctors Ted Bryce and Greg Dusor, as well as people in their lab, helped him develop the calcium imaging of these pain neurons. And that work was done in their lab. And he then enlisted many of his neuroscience questions to folks in the neurosciences lab. So this is a combination of good old shoe leather microbiology as well as cutting edge neuroscience. And then Tian Quinn and his lab synthesized the S1 precursor molecules. So he had the chemist involved in making some of the molecules that they desperately needed to definitively test their hypotheses. And then Lauren Gatron and Dr. Michael Burton isolated and cultured the primary ganglia that they used for these assays from the human members. So again, it really took a village to accomplish all that went on. But, none of this would have been possible without the microbiology. And that is his lab members who on a day-to-day basis helped him work in the biosafety level 3 lab. Those of you out there who work in BSL-3 conditions know how fatiguing it can be. And Harris Kahn and Brianna Pascoe greatly helped Cody on a day-to-day basis conducting these experiments and actually doing the viability assays that he had to do to culture to demonstrate the pathogenesis of the mutant TB that they were growing that were not making this particular lipid. So Cody acknowledges that every author listed on this paper was incredibly helpful to him, and he feels their contributions were all significant. So again, he's sharing credit with his coworkers, and it took a village to accomplish this, but now we have a better understanding of what's actually going on when you hear the phrase, it feels like I'm coughing up a lung. If you've ever talked to someone who's had active tuberculosis. I first learned that expression from an old infectious disease doc when I first came to South Carolina, who got tuberculosis working in the emergency room as an intern. And he's the one who taught me how highly infectious it was, even despite the best precautions they would take. And so I asked him, I says, well, what is it like to have tuberculosis? And he says, it feels like you're coughing up a lung. And it really, this cough reflex induced by this now simple sulfatide lipid that when we talk about virulence factors, when we introduce TB to medical students or dental students, we always talk about the sulfatides. No one ever knew what they did. Now we know they make you cough up a lung. So Cody really has added to the literature. And if you can say that as a graduate student and get a paper and sell, so much the better.
Yeah, and it took persistence. But I also thought it was interesting that Cody really recommended to his other colleagues that they network and that the ability to talk across disciplines and collaborate with a wide variety of people, really, the value of that really came through in this paper.
Oh, yeah.
You never know where the biology is going to take you and who you're going to need to help you out. So build that broad network of smart scientists.
This it's interesting, the choice of a guinea pig model. I wanted to comment because guinea pigs cough, but mice do not. And so if you're wondering why they don't use mice, I don't know about TB growth itself, but mice don't cough, neither do they vomit. So mice can't do a lot of things that we would like to study.
But Mickey Mouse can cough. I've seen Mickey Mouse cough.
I was hoping that one of you would be able to do a guinea pig cough for our audience.
No.
Is that what they sound like? I was imagining it was like.
It's perfect. It's perfect. That's very cool. So, Michael, this idea of coughing up a lung made me think. I was talking to Christian Drosten a couple of weeks ago about coronavirus, and they did a study with patients where they recovered virus from the upper and the lower tract, right, all the way down in the lung. And they found a single nucleotide polymorphism, so they could track virus, and they found that only virus from the upper tract is transmitted to other patients, not from the lower parts of the lung.
Well, that's where TB grows.
Now
that's where TB grows.
It only grows in the lower tract. Is that correct?
TB grows in the upper.
Okay.
Well, the upper chamber of the lung. I don't know that it grows in the respiratory tract.
No, it doesn't. It grows in the upper chamber of the lung. That's where it is on the X-rays.
So the point is that for virus, the upper tract titers of SARS-CoV-2 are very high, and that's a very easy place to expel, droplets. Oh. I think it's harder to get things out of the alveoli out into the world, right?
Yes.
So that's why this coughing up a lung thing sounds really interesting, but I don't think you actually expel things from your alveoli regularly. I don't know how far down your lung you can actually expels something. I'm sure people know, but certainly the upper tract, it's right there. Saliva, as we just saw, and nasopharyngeal droplets for sure. For sure. This is very interesting, Michele, the whole cough idea. I think people don't really do this very much for viruses.
Well, they're hard experiments to do, and they're not safe to do if you're working with a pathogen.
And of course, you have to have an animal that will cough, and ferrets do cough, right. Although the paper we did last time with SARS-CoV-2 showed that the virus does not induce cough in ferrets, so can't do it there. Maybe guinea pigs.
Well, if you sneak the gene for the sulfatide into the SARS-CoV.
Well, I was thinking of that. Maybe not SARS-CoV-2.
Yeah, please
maybe
don't.
Something else. It's probably not one gene, right? It's a pathway.
It's a biochemical pathway. The virus is not big enough for it.
Hey, what are you saying? It's pretty big, 30,000 bases.
Yeah, but that's a whole biosynthetic pathway.
Yes, I agree. All right. Thank you, Michele.
My pleasure.
I want to read one email before we wrap up here from Harry, who's from Australia.
Melbourne.
Big fan of the show. Great to hear such enthusiasm from such experts here in Melbourne, Victoria, Australia, rather than Arkansas, California, Florida, Iowa, or Kentucky, where there are also Melbournes. It's 13C and drizzly. Currently working in clinical micro. I wanted to write in about PCR and serology testing for COVID-19. Hey, look at this. He wrote into TWiM. First, about PCR. This is undoubtedly the most valuable test modality. While RT-PCR is a remarkable technique capable of detecting tiny amounts of RNA, does not mean the commonly used test is highly sensitive. It seems the sensitivity of a SARS-CoV-2 real-time PCR on a single nasopharyngeal swab is about 70%. That is a false negative rate of 30%.
Wow.
One explanation seems to be that COVID-19 patients only intermittently shed in their nasopharynx. PCR performed on sputum when patients are producing sputum seems more sensitive, while there's where the saliva might be better off.
Yeah, it is.
An overriding difficulty with examining these new tests is the difficulty of defining a diagnostic gold standard, given this is an entirely novel disease. I've heard from Chinese infectious disease doctors that initially in Wuhan and Shanghai, the gold standard rule out of COVID-19 was 3 negative PCRs over 3 days, with 1 performed at a reference lab, then no SARS-CoV-2 genetic material detected by metagenomic sequencing on nasopharyngeal swabs and or pulmonary aspirates.
Wow.
Oh my gosh, you can't do that now,
That's expensive.
Right? No, that's expensive too.
After all, the only initial tool was metagenomic sequencing because no PCR had been developed. I've heard that many patients were found who had a typical illness pattern but repeatedly negative PCRs before SARS-CoV-2 nucleic acid was discovered through metagenomics, clearly not affordable in most health systems. The language used in the media is confusing regarding testing and seems to assume that the PCR is perfect no matter what circumstance it's being used in. When multiple of Donald Trump's contacts developed COVID-19, he released a statement saying he tested negative and had no symptoms. This shows a fundamental misunderstanding. A negative nasopharyngeal swab PCR means precisely nothing in an asymptomatic person. It neither rules in or out current infection, and it certainly gives no predictive value about the probability of developing illness. If Donald Trump developed symptoms the day after being tested negative, he should have been isolated and undergone another test because he'd presumably have developed COVID-19. Good luck to anyone who had to explain that to him. If a patient in New York or New Orleans now has fever, cough, loss of smell and taste with a bilateral pneumonia, then they should absolutely be diagnosed with COVID-19 regardless of any test. The pre-test probability would be so high that even with a negative test result, COVID-19 is the most likely diagnosis. That's a really good point.
It is.
If testing capability is limited, it's therefore entirely reasonable to not test people with a typical cluster of symptoms and signs. They should just be diagnosed and managed as COVID-19 patients, and testing resources used to discriminate other cases where the diagnosis is less clear.
And that's what China did there once the epidemic was in full roar in Wuhan, is they all of a sudden revised their definition based on clinical symptoms in order to continue testing the naive, the individuals who were not displaying symptoms. So that strategy sounds reasonable to me.
And then he has some interesting comments on serology. It has no role in testing for COVID-19 currently. It's tempting to consider the presence of antibodies to the virus to be definitive evidence of past or present infection, but this assumption has many holes. First, IgM assays are notoriously nonspecific. Mostly for this reason, there's almost no disease that can be definitively diagnosed by the presence of IgM alone. IgG assays are more specific for the antigen. However, they can take weeks to develop. In any one assay, there will always be a false negative rate on an IgG test. For whatever reason, a previously infected person may not have antibodies against the particular antigen being measured by the assay. Unfortunately, currently the presence of an IgG against SARS-CoV-2 is not evidence of immunity from reinfection. That's right. It's not even evidence that viral shedding has resolved. We just don't know either of those things with enough confidence for COVID-19. While I think it's a solid guess that past infection will give some degree of protection from future disease, we certainly cannot assume this will be complete immunity. We therefore can't offer any different guidance regarding PPE, removal from isolation, or return to work for anyone on the basis of a serology result. You are absolutely spot on. Go talk to our leaders. Public Health Laboratory Network of Australia has released a statement on point-of-care serology tests, which is freely available, and he provides a link and has a question. My question relates to antiviral therapies. Which mechanism in the SARS-CoV-2 cycle do you think is more likely to yield a useful antiviral drug? And I would say the enzymes, the polymerase and the proteases, because you can easily target the active sites and there's not as much of them as any other protein. And those have been the most commonly targeted enzymes. If you look at all the antivirals we have about 100. Most of them are against enzymes.
And that's how we've licked hepatitis C with the protease inhibitors.
That's right. Protease and polymerase.
Yes.
And HIV too.
HIV too.
Polymerase and protease inhibitors. So that's what I think. And Harry ends all the best in hand washing and distancing. Harry is a trainee, microbiology and infectious diseases, advanced trainee, and a microbiology registrar. Wonderful email, Harry. Thank you so
thoughtful.
much.
Lovely.
Really clear. That is just great. All right. That is TWiM 216. You can listen on any podcast player. You know, if you're home and you're bored, just listen to all the back catalog. You know, it doesn't go out of date. 216 episodes. Like an hour each, that's a long time. That's many days that you can alleviate your boredom. So listen in on your podcast player. And if you want to see the letters in the show notes, microbe.tv/TWiM. If you like what we do, consider supporting us financially, microbe.tv/contribute. And of course, we love getting your questions and comments, TWiM at microbe.tv. Michele Swanson is over at the University of Michigan. Thank you, Michele.
Thank you, and stay safe, everyone.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Well, it was my pleasure. I learned a lot today. Thank you.
Yeah, me too. Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I am Vincent Racaniello. You can find me at virology.blog. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.